Hi, Dr. At.
Yeah.
I'm also a student of Dr. Solez's. However, I don't possess any formal medical education, but I've worked enough with Dr. Solez to be a little familiar with this topic. And from all the literature that I've read about regenerative kidneys, it seems that the kidney is like really the, I guess, holy grail of regenerative organs, just because of the complexity of what's inside it, right? And my question to you is, when you're seeding these scaffolds, uh, how are you guys ensuring that the cells that make, for example, the loop of Henle, how can you ensure that those cells end up in the right place? Because with all the literature that I've read, it seems that they just kind of put the cells in, and the cells have kind of like a mind of their own, and they just happen to turn into the correct system.
No, I think that's the key question, right? So both actually kidney and lung have the same issue, that you have a relatively large epithelial compartment that needs to be seeded with a site-specific phenotype, and we just don't we haven't figured that part out yet. The question is, how much of it do we need to figure out? So what we saw in kidney reseeding was that glomerular epithelial cells tended to stuck more to the glomerulus, right? Podocytes, and think that is a little bit of a niche effect based on the integrin profile of the glomerular basement membrane. But to say that distal tubular epithelial cells stuck to the distal tubule and proximal tubular epithelial cells stuck to the proximal tubule is not right. I mean, the issue is that we see that a mix of tubular epithelial cells onto the loop of Henle as a whole, basically, and whatever stuck, and did not get to the point where we're able to distinguish the formation of proximal versus distal tubule. The question is really, from a device perspective, how much do we need, right? How much of a functional nephron do we need to regenerate to get somebody off hemodialysis? I mean, basically, an organ would need to be blood perfused. So blood needs to go in, blood needs to come out, and there needs to be filtration somewhere in between. And I think with a completely re-endothelialized glomerulus, we're already relatively close to that. And then the next step is, of course, the myriad of advanced functions that the tubular epithelium of different location fulfills. The question is really, how many of those do we need? If you think of hemodialysis, there's not a single cell in hemodialysis, right? There's two fluids and a membrane in between, and it works okay. It doesn't work great, but it works okay. It keeps people alive for a while. And so I wonder if we can take that approach to say, what are the minimal design criteria for our epithelium? What are the minimal things that this epithelium needs to do? Do we really need concentration, or do we really need active, let's say, bicarbonate production? Or can we get away with less, basically? And I mean, to get back to Dr. Solez's argument from the beginning that there might be synergy with current existing industry, I think that's one of the selling points, right? Where you say, well, you're looking at somebody with a whole different animal of end-stage renal disease. You're somebody who can get rid of fluid but cannot control his salt metabolism well, or cannot produce erythropoietin, for example, or cannot do X, Y, and Z. And so that person becomes suddenly a surviving customer for you, or for pharmaceutical industry in that sense, with an imperfect organ. There's a winded response to your question that we don't have a good method yet to get the right epithelial cell in the right location, absolutely. And I think to believe that it will all magically happen is wishful thinking. I don't necessarily think so. Because the phenomenon of structure formation from scratch is highly complicated, right? And temporal spatial control of growth factors, etc., that we just cannot replicate at this point.
In terms of your trials, has there been anything that you guys have noticed that you can get away with?
So we haven't done enough large animal trials yet in terms of transplanting regenerated constructs to see how their long-term function is. What we've been working on for the most part was to regenerate an iPS-derived vascular system over the last 3 years that can support a blood perfused graft for a longer period of time. So we're still at that stage.
Okay. All right. Thank you very much.
No, thank you.
So I've been thinking about the kind of big data aspects of this, that if you take a decellularized organ, and if it works pretty well as something you can seed and generate a tissue engineering organ from, if you could somehow simulate that, if you could figure out every parameter of the very complex shape and so on, and fabricated it, then you could make, you wouldn't have to have a source of these organs to be decellularized. You could just make them, synthetically from that first template. Are the number of parameters there beyond what a machine could ever handle? I don't think so. I mean, once we figured out what we know about what extracellular matrix components need to be there, and everything that's known about what a successful template is, then we could just reproduce that rather than having to find an actual organ to flush the cells out of. Do you think that that's a possible way in which this would work in the future, or are we always going to need a sort of biological source for the organ that we're going to put the cells into?
No, I think, the only correct answer to that must be no, right? We're not going to always need, because,
right.
Seeing how technology evolves, we'll figure it out at some point. I think right now it's just tremendously hard, and that's when I came from cell therapy and wanted to do a little tissue engineering, basically, that's, I used to inject muscle cells into the heart. That was my job and my interest, and I realized it's not really going to regenerate functional heart muscle, simply because the structural impact of that injury of a heart attack is too big. And so I went to tissue engineering, and there wasn't a good way of creating scaffolds at the time. I mean, there was electrospinning, and 3D printing wasn't around. I mean, it was invented, but stereolithography was invented, but wasn't really applied to tissue engineering yet. So there are techniques or were techniques of synthesizing a matrix. It's just that they weren't there yet, and I think that's still where this is.
Right.
If you look at the alveolar basement membrane, you're looking at a elastic fiber collagen membrane that is less than a micron thick, right? Mm-hmm. And that at a three-dimensional complexity and density of vascular channels and interconnectivity, that's just hard to replicate with current technique.
Yes.
And I think that taking a pig lung or a pig heart or a pig kidney and perfusing it with detergent is a very simple and cheap thing to do. The pig costs you $300, and the kidney explant time is 30 minutes on a clean table, and the perfusates overall cost you maybe $200-300, and so you have a decellularized scaffold that is the ultimate platform for under $1,000 for a human-sized organ, versus creating the complexity of this tremendous complexity on a human scale will be very time-consuming in a very sophisticated machine.
Yes.
Require a lot of very complex technology. Not to say that that's not where we should be going and will be going, because of course, what we deal with is the perfect organ. But if from a device perspective, again, we might get away with less complexity. There's an argument to be made to try something less complexible, like a dialysis membrane, for example. So I think that, and that's part of the reason why we do a lot of compositional and architectural analysis of these matrices to say, well, what would be the components that we'd have to put together, right? What would we make this thing out of if we would like to replicate, let's say, a postnatal week 1 lung or kidney or whatever. It's just that I think it's very complex. So technology, I think, will be there at some point. It's just not there right now. And what we're focusing on are different questions. So we keep using the old platform, basically, because it enables us to understand how to guide cellular morphogenesis and how to direct this these later stages of development, while we don't have to worry about matrix. And if somebody comes along and makes an artificial matrix, I think these technologies will apply. We'll be able to apply them to that different scaffold.
Right. Well, as you know, stem cell, the field of stem cells in general has very positive things, but it also has stem cell hype, where people predict that advancements will be made more rapidly than they will, and that sort of thing. So without wanting to feed into that problem, I think there's a kind of advocacy within transplantation and renal medicine and probably other fields that needs to be undertaken to give the people in that field the idea that they're not that different from the regenerative medicine folks, that and we could use their help. It isn't like that when the AST created the regenerative medicine community of practice, that everybody else could heave a sigh of relief because now they don't need to think about this stuff anymore. That small group of people in that community of practice will just take care of everything, and 10 to 15 years later come back with all the answers, and they don't have to worry about all this silliness about stem cells and so on. But of course, that's not really true, that without the help of the mainstream people in the transplant field, progress is going to be very slow. And if there are few people working in this field, if they're regarded as a real niche field in several senses of the word, then progress is going to be exceedingly slow. So really, I think without telling people this is all going to be there in 5 years, we need to create a kind of excitement about it. Right. And a kind of, inclusiveness, so that people feel that their existing skills, particularly what they learned in medical school that they may feel is not all that valuable to them anymore, suddenly these things are important enough, important again. Like, I think of Neil Bricker's intact nephron hypothesis, for instance. It was a big part of one's learning of renal physiology back in the 70s. Suddenly, it's important again when, some tubules have cells, others don't, and it's right, how would a kidney configured in this way work? What's the background to help me figure that out? So that the things people learned in school that they may have thought, although they may have, nostalgia for it, that they'd never use the information again, we could actually use the information that they have, the specialized knowledge that they have from the past, to solve some of the questions that we're now facing with.
No, absolutely. I mean, I think the key to this is, and that's always the problem, of course, with forward-looking statements, right? Like, if you make too many forward-looking statements, everybody eventually says, well, you're a lunatic and you're just trying to get PR and trying to get attention for something that's never going to work. But the benefit of it is that you show people a platform. Instead of being the lunatic out in the garden, you basically legitimize the effort. Right? Yes. You legitimize the topic of organ regeneration in a way that they can apply their skill set to it and help advance it. Because that's what's fascinating about my lab, the research I think in my lab that I find fascinating when I walk into it, because I'm a thoracic surgeon, right? Mm-hmm. But there's people who are developmental biologists, stem cell biologists, physiologists, bioengineers who work together and apply themselves to this crazy topic of the the lunatic out in the garden trying to make the organ regeneration happen, and that. I think the effort to explain to people that this is something, because of its potential impact, is worthwhile pursuing, will help us pulling in these different disciplines. For example, with lung, the lung paper at the time, Ewald Weibel was one of the people who helped me. He's a pathologist who really quantified how many alveoli are in the human lung. He used to work in the States, then went back to Switzerland. He's long retired, but I came across one of his chapters and sent him an email and said, "Can you help me? I'm looking at this completely acellular lung now. Can you help me characterize that?" Or another example is that there are papers from the 70s looking at freezing injury in rat kidneys and how reepithelialization occurs, and how it's interesting that it occurs if you freeze but not if you burn, right? Well, it makes a lot of sense. I mean, freezing is nothing but decellularization. So interesting that you mentioned that. That I think there's a lot of disciplines that need to apply themselves to this kind of research, and it's our job to convince them that it's not completely crazy.
Yeah. Well, let me tell you some of the pitches that I make, particularly surrounding your research. So I go into a room with my computer and a PowerPoint and so on, and I show the nature video first, and then I ask them to imagine a world where the person doing the most interesting kidney research is actually not trained in kidney medicine primarily. Can they imagine this? Of course, all these kidney physicians sitting around, and they realize this is what they just experienced, that whatever they're doing is less exciting than what they've seen in the video, but they've spent substantial portions of their life learning nothing else beyond the kidney, and you probably haven't. So I think that's bound to be stimulating for them, because if you look at the converse of that, how could somebody who actually knows about other parts of the thorax and knows about the heart and the lung and so on do such interesting work? Well, doesn't it mean that they could help you succeed faster? And I think it obviously does.
Right.
If they've been spending their entire career focusing only on the kidney, if you're trying to make a stem cell generated kidney work, these are the people who could help that effort succeed more quickly, and it would be satisfying for them and for you. And the same argument works with every other specialized organ. And it seems to me that looking upon any part of this as a threat sort of does a disservice to the patient, right? Because if we're only serving 10% of the patients, we're talking about serving 100% of the patients. Any perceived threat going from 10% to 100% is counterbalanced by the amount of good we're doing for the human race by scaling things up like that,
Right, and it's also, I think it's a development. It's in one way it's a platform to develop a clinical tool from technologies that might otherwise not necessarily have enabled that, right?
Yes.
Say, tubular regeneration based on cells, or looking at interstitial cells. I mean, a lot of these research areas, one could apply to this platform to translate their knowledge into a clinical application. But also, I think this platform could be an interesting development field for them, right? I mean, it is a test bed where you can look at cell potential, you can look at the tissue formation, you can look at cell-cell interaction in a very controlled and isolated fashion. So that collaboration not only is I think, or that cross-pollination is not only interesting because it applies their science to our problem, but it potentially helps other areas to make new discoveries, right? Like look at the potential, for example, in lung, we look at the regenerative potential of basal cells that had been a big topic in a bunch of Nature papers last year. And we're looking at how these cells, human cells, actually do engraft and regenerate airway and alveolar epithelium, or do they not? And this versus the mouse models enables you to really take human basal cells and put them on an empty basement membrane and see if they do it or don't. It's a very clean model. I mean, if there's no surfactant producing cell, well, there was no distal differentiation there. So I think it's a model system in itself that can be applied to many different areas, and unfortunately not everybody sees it that way.
Yeah, but there's some intrinsically exciting things, I think. If you think even about exciting young people about this, say going into high schools, stuff like that. Like the color change. You take an organ, you decellularize it, and it turns white, and you put in stem cells, and it turns pink again, and then maybe it starts to function, and so on. But I think what they could also understand is another way to turn it pink is to just infuse one kind of cell, right? One kind of completely differentiated cell, and will still turn pink, but it won't work because those organs are not going to function with just one kind. So there's a whole lot of complexity that at the macroscopic level you don't really appreciate, where suddenly the microscopic milieu becomes very important, but you can't get too hung up on needing to reproduce normal structure 100%, because obviously every organ will work probably with some figure, whatever it is, I don't know, 79% reproduced, original cell configuration or something, or 50% or something. We'll end up finding out. And it will be exciting, actually, to see how well organs that we know have those anatomical defects, how well they actually function. I think that's part of the exciting
right.
thing.
And I think it's important to keep an open mind there, right? I mean, who would ever have thought that you don't need pulsatile blood flow to survive?
Yeah.
Well, I mean, the first, the thoratec extracorporeal ventricles, they pumped blood in a pulsatile fashion because that's what, it was easier, I think, to build the pump that way, but maybe it was, but it was also thought, I think, that pulsatility matters a lot. And then the first left ventricular assist devices came out without any pulsatile flow. And, you have side effects from it, but you survive with it. So it's interesting, like you say, to find out how much complexity do we need and what is actually required to, because what we're up against is I mean, the bar is not all that high, right?
Yes. Yeah.
Hemodialysis or ventricular support or extracorporeal membrane oxygenation, I mean, those are non-physiologic replacements. So,
yeah. Well, I want to thank you very much for taking part in this session today.